All right. Now, let's start with the head and then go all the way down through the various regions. Cranial neural crest cells. Here are the fates of the cranial neural crest cells. We have cartilage and bone, which are typically mesoderm derivatives. So this is, again, this is something that's just interesting about neural crest cells, is they originate from ectoderm, but they can become cartilage and bone, which are typically mesoderm, as well as connective tissue, which is typically mesoderm. Now, cranial neurons and glia, these are neural derivatives. So here again is the multipotency of the neural crest cells. There are relatively little to no Hox genes being expressed in these regions where cartilage and bone are being expressed. They found that trunk neural crest cells have the potential to become cartilage and bone, but under the influence of Hox genes along the trunk region, it suppresses the ability for these neural crest cells in the trunk to become cartilage and bone. So in reality, neural crest cells all along the axis of the embryo have a huge potential, but certain regions may suppress that ability due to gene expression. So that's one of the reasons they found that cranial neural crest cells can actually become cartilage and bone, whereas trunk neural crest cells cannot due to the Hox gene expression. So let's look at some of these aspects. Now, I talked about the rhombomeres. Rhombomeres are kind of these discrete regions of the hindbrain that these neural crest cells are forming from. What's interesting about the rhombomeres is they typically migrate on the even rhombomeres, rhombomeres 2, 4, and 6, but they don't typically migrate from the odd rhombomeres. Now, cells still form in the odd rhombomeres, rhombomeres 3 and rhombomeres 5, but they don't have the ability to migrate from those. So where do they go? The ones in the anterior rhombomere 3 will actually enter into rhombomere 2 and migrate out that way. And the ones in the posterior here in rhombomere 3 will migrate out through rhombomere 4 area. So you'll actually see them splitting and going out in those ways. Very similar to the splitting that you see around the somite, but there are no somites here. And so there are, again, signals here that restrict the migration of neural crest cells out of these regions and cause them to mix in with the rhombomeres which are adjacent to them. Now let's go through some of the derivatives. One of the things that is not always clear in your book, and I want to help clarify, is they'll talk about rhombomeres and then they'll talk about pharyngeal arches, and they are not the same thing. Let me show you the difference between the two. Here are the rhombomeres. The rhombomeres form in the dorsal part of the neural tube where the neural crest cells form. So here are the rhombomeres right at the most dorsal region of the neural tube in between the ectoderm and the neural tube itself. The pharyngeal arches are these. This is where the cells from the rhombomeres migrate to. So when the book says, oh, the derivatives of the pharyngeal arch 1 are this, actually, the first pharyngeal arch has a combination of cells from multiple rhombomeres. Okay? So just keep that in mind as you're reading through the book. I'm going to keep it pretty simple as we go through here in terms of what some of the fates of the cells from each of these rhombomeres are. Again, you can see from rhombomeres 3 and 5, these typically have to move outward and go to rhombomeres 4 or 6 in order for them to migrate. Okay? Now, I'm not really mentioning some of the later rhombomeres or the first ones. We're just going to focus on these middle ones here. Let's look at that. So what are some of the things that they come from? Well, some of the bones, in fact, the great majority of the bones that form in your head come from neural crest cells. You have multiple types of bone formation. So I'm going to spend a little time talking about how bones form. Most of the bones in your head are either irregular or they're these flat bones. And the way in which they form is different than how your long bones form. Your long bones form first by filling out this kind of cartilage matrix and then filling it in with bone cells. Well, that's not the same way that the head bones form. They typically will undergo what's called intramembranous ossification, which is where the cells will start forming and then just start forming bone right away. They don't form this cartilage model and then start forming the bone from that. They just start forming the bone intramembranously or in the tissue itself rather than setting up a model. That's the big difference between how you get endochondral ossification, which is how your long bones form of your limbs, versus the flat bones and some of the irregular bones. Let's look at some of the bones of the head. The malleus and the incus and the stapes. Where are those found? The ear. Those are the inner bones of the ear. These come from neural crest cells. So the inner bones of your ear that determine your ability to hear, that join up with your cochlea, these originate from rhombomeres 2 and 4. So the malleus and the incus come from rhombomere 2. Rhombomere 4 forms the stapes, as well as some of the other bones, the styloid process, the hyoid bone. The styloid process is at the base of your skull where you have a connection for tendons and things of that sort. So these are just some of the derivatives. Again, a lot of the bone that you're going to have in your head as well as your inner ear, these come from the rhombomeres. One of the things I want to illustrate is that up here in rhombomere 2, there's actually no Hox genes whatsoever that starts forming some of these malleus and the incus. There are some Hox genes here in the further rhombomeres, though they still have the potential to become bone and cartilage at this point. It's when you get further down and start getting into the trunk region that other Hox genes, the later Hox genes, will start to prevent these trunk cells, neural crest cells, from becoming bone. Here is intramembranous ossification. Essentially, these neural crest cells will migrate into their respective regions, and then they'll take on a fate where they'll start to become osteocytes, which are the bone cells. They'll start forming a calcified matrix, and that's what's going to start forming a lot of the flat bones that will form your skull. Remember, there's a difference between rhombomeres and pharyngeal arches. This table just illustrates the 6 pharyngeal arches, which are these regions right here, and what they eventually become. Really, there's a large mixture of these neural crest cells that will come into these pharyngeal arches. And so therefore, you're going to see a lot of differences or derivatives of these various regions. So here just illustrates pharyngeal arch 1. You got the malleus and the incus, Meckel's cartilage, and you got some facial bones here. And then you have pharyngeal arch 2 and 3 and 4 and 5, and it just respectively shows the various bones that will come from those regions as well. Okay, so there are other cells that come from the cranial neural crest cells that will form neurons and glia. We'll get to those here in just a little bit. Okay, though we'll talk about those when we get to the next stage, when we talk about some other types of neurons. So this just shows a lot of the cartilage and bone that will form, because that's fairly unique to these cranial neural crest cells that you won't find in other regions. Let's talk about cardiac. These are actually the simplest of the 4. Cardiac neural crest cells have a number of fates. They can become melanocytes, they can become cartilage, they can become neurons, and they can become connective tissue. Okay, so there's a number of different things. They form melanocytes near the heart region. They'll form neurons that are associated with the heart. They'll form cartilage, another type of connective tissue. Some of the more important connective tissue that the cardiac neural crest cells form is they'll actually form the connective tissue of the large artery, as well as the septum between the branches in the heart. So we know that the heart primarily forms from the mesoderm. However, we've shown that the cardiac neural crest cells play a key role in certain parts of the heart as well. So even though most of the heart forms from mesoderm, cardiac neural crest cells are mixed in as the heart is forming and helps to form the septum as well as the arteries that come from the heart. So they play a key role in that generation. Here it just shows an example. They're really restricted to between somites 1 and 3. So it's just a small little area where these cardiac neural crest cells will form. Okay? And as they migrate towards the heart, they're going to take place in a number of differentiation processes. They're going to form neurons that are associated with the heart. They're going to form the septum and then arterial muscle wall. There will be some melanocytes that they form in the skin around that particular area where the embryo is developing. So there's a wide number of fates of these cardiac neural crest cells. But it's very simple, and they're very restricted to that particular area.